So, this is our first sort of entry into thinking about structure of biological molecules and how that structure affords function. So, for each of our four learning units that cover those four biological molecules, we've got nucleic acids, we've got proteins, carbohydrates, and lipids. Remember, at the end of our first learning unit, we sort of wrapped up with sort of this 30,000 foot view of the different biological molecules we're going to study. So we are going to pull this sort of sheet back out every time we start a new biological molecule and remind ourselves of some of the high level things that we need to think about. So all of these are going to have linkages that we're going to talk about. So when you see me do this, I'm talking about linkages of either monomers or different functional groups that allow us to form an important linkage that's going to be relevant for that biological molecule. So for nucleic acids, the linkage is a phosphodiester bond, and that is going to hook nucleotides together to make nucleic acids. So here's some basic sort of structural information that we'll continue to build on in this learning unit. And importantly, with all of our biological molecules, we will address four different functions or four classes of functions that we might think about. We'll think about the ability to participate in information storage. We'll talk about the ability to store energy. We'll talk about the ability to perform catalysis. And importantly, we'll talk about whether or not these biological molecules have a structural function to them. So specifically for nucleic acids, of course, we think about nucleic acids carrying information. That's our genetic code. We will talk about the central dogma. We'll talk about the omics of nucleic acids, whether it's genomics, whether it's transcriptomics with messenger RNA. It's proteomics with the protein that's translated from that messenger RNA, or we're talking about the metabolites that are produced from functioning enzymes that are proteins. So again, another video that'll be out that talks sort of about the omics piece of this, but that's going to be an important piece. Our biological money is a nucleotide, okay, adenosine triphosphate. So we'll look at the structure of that. We'll understand why it's high energy in our first learning unit, right? We talked about the fact that it is worth 30.5 kilojoules per mole, and we looked at it from a reaction coordinate standpoint to understand why our biological money is valuable, why it's stable, but then how we access it. So again, learning unit 1 sort of dealing with ATP there. When we talk about nucleic acids as RNA, right, we're going to have messenger RNA that's part of the central dogma and information piece. We're going to have ribosomal RNA that actually performs a catalytic role within the ribosome. So that is the protein synthesis machine in your cells. And then we're going to have catalytic RNAs that are ribozymes, so another sort of catalytic piece of RNA. But when we think about those three types of RNA that we're going to just touch on just slightly here, there'll be messenger RNA, ribosomal RNA. And then I put the transfer RNA piece sort of from a structural standpoint here because it is the structure of unique transfer RNAs that code for certain amino acids that allow us to take DNA, transcribe it into the messenger RNA, and then that messenger RNA has a 3 codon piece that matches up with 3 nucleotides that are on our transfer RNA that bring in that cognate amino acid that are going to allow us to make the proteins. Now, I know that was a lot, right, in that 30,000 foot overview. Those of you who have taken cell and molecular biology, or you have or will take genetics, a lot of those processes will be covered there. We're really looking more at the biochemistry aspects of nucleic acids. And to that end, with all of our biological molecules that we'll study here in different learning units, we start with thinking about what the structure of these molecules are. We always start with thinking about the monomer form, and then we address how do we link things together, and then we see function and application. So that's how all of these learning units are going to be set up. So only 2 slides here in this sort of first section, thinking about the structure. So I'm going to talk a little bit about what the building blocks are, what the monomer building blocks are for nucleic acids, and those building blocks are nucleotides. So when we think about nucleotides have 3 pieces to them. They're going to have a base, they're going to have a sugar, and then between 1 and 3 phosphate groups. And that has to do with whether they are a nucleotide monophosphate, diphosphate, or triphosphate. So before we get into how all 3 of these pieces are together, I want to take a minute and sort of highlight each one of them sort of individually. So when we think about our bases, we think about these are called nucleobases. So these are nucleobases. And they come in basically two flavors. They come in a 2-ring form, which we call purines, and then a single ring form, which we call pyrimidines. And pieces that I need you to know is that these are modified with different sort of exocyclic groups that allow them to make the 5 nucleotide building blocks that we see in both RNA and DNA. And if you are of the purine nucleobase flavor, that's how we make our A's and our G's. And then pyrimidines are going to be our C's and our T's that we see for DNA. And then I'm going to put U there because that is what we see for RNA. So these are the nucleobases. You don't need to know structures of them. I just want you to be able to recognize that purines, which are the 2-base form, is what we see for A's and G's. These are the 4 letters that we essentially build our genetic code from. And then when we talk about pyrimides, that's C's and T's. I will point out here, because the numbering will become relevant later, when we talk about the nucleobases, the numbering is just regular numbers, 1 through 6 in a single ring, and then 1 through 9 if we've got a 2-ring system. That's going to become relevant because when we start talking about sugars, you're going to hear the word number with prime after it, and that has to do with the numbering in the sugars. So let's go ahead and take a look at then the sugars that we have. So again, bases plus a sugar are going to be what we call a nucleoside. So these two pieces together are a nucleoside. Okay, and we have 2 different flavors of sugar that we can see. So in green here, I'm going to highlight, here's our different sugars that we have. I'm going to zoom in here so we can see this a little bit better, because the sugars are a piece that I want to talk a little bit more about the structural details of. We're going to see these sugars again when we have our carbohydrate learning unit. But here is the sugar that we see for RNA. Here's the sugar that we see for DNA. And I'm going to highlight the main difference that we have here. The main difference is what we have highlighted in green here. We have the sugar ribose. It's a 5-carbon sugar that we are going to see when we have RNA. When we have DNA, that D in the DNA actually stands for deoxyribose, and that means we don't have the oxygen at that position. And specifically, this is the 2' position. So we'd say that it's 2' dehydroxy ribose. And in this case, we're going to be talking about some of the structure function sort of relationships that make RNA different, because at the 2' position, it has an OH group. Okay, so that's going to become relevant later. But I want to highlight some other important pieces that we have here. I'm going to highlight that at the 3' and 5' carbons, we also have hydroxyl groups. Those are not the only places that we have hydroxyl groups, right? But these are 2 that are important, because these are going to be the points for linkages, okay? These are going to be the points where we can add phosphates, and then when those phosphates are put between nucleotides, that's how we form our phosphodiester linkage. So we're going to see that again in a little bit. Okay, so base plus a sugar gives us a nucleoside. When you start adding in phosphates, and again, you can have 1, 2, or 3 phosphates, this is how we make a nucleotide. So highlighting here, when we have all 3 pieces here, okay, this is going to be a nucleotide. So a nucleotide, and maybe this little T here between nucleotide and nucleoside can be a reminder that you have 3 parts. So that T can remind you for 3 parts. And reminding you, those 3 parts are, we're going to have a base that's attached to a sugar. And I'm going to show you in just a second how we see those pieces connected. And then anywhere from 1 to 3 phosphates. So all 3 of those we need to have in order to see our nucleotides. Okay? So in this box that we have up here, right, or in this section here, we're going to see the different nucleotides. And we're going to see a ribonucleotide and then a deoxyribonucleotide. So I'm going to zoom in here and sort of remind you of the different pieces that we have here, right? We said our base was going to be in blue, right? So there's our bases. All that I need you to know is where the base attaches. I don't need you to know the structure of the bases, but the bases are going to attach at the 1' position of these sugars. Okay? Again, the sugar is going to be different depending on whether we have a hydroxyl group in the ribose sugar or a hydrogen if it's in the deoxyribose. And then importantly, we can have phosphate groups attached at different positions. You can have phosphate groups that are attached at the 5' position. This is typically when we see things like ATP. It's going to have 3 phosphates attached at this 5' position, but you also can have a 3' phosphate group, okay? And so one of the things that we're going to see is I do want you to know what this phosphate group is. So this again is a phosphate group. It looks like this, right? So a phosphate group, it's going to look like this. This is our point of attachment, okay? In future, we will abbreviate it just a P with a circle, but I'm going to highlight here. I'm going to show you how we want to be able to draw a dinucleotide. This is a skill that you're going to need to know for when we draw molecules for not only our regular exams, but this is going to be something you're going to still need to know how to do for your final exam. It's always going to be a dinucleotide, so I'm going to show you how to draw a generic dinucleotide, and then I'm going to show you how to map on for an example of a code that I would give you for a specific dinucleotide, how it would change that base structure. Okay. So what I'm going to do in this blank space that's over here is I'm going to draw that generic dinucleotide. We're always going to draw them in the same way. They can be shown sort of in different orientations, but if we always draw them in the same way, that'll help us remember how we're going to do that. So generic dinucleotide is always going to start with my 5' hydroxyl. And then I'm going to have that come down to a ribose sugar. So I'm sort of mimicking what we have here. I'm going to draw this exclusively as a skeletal structure. So this has to be shown as that little bend there. We have to make sure we don't lose that carbon atom. Here's our 5-carbon ribose sugar. Remember, at this 1' position is where we're going to attach our base. Remember, this is going to be a generic dinucleotide right now. Depending on whether it's DNA or RNA, it's going to have a hydrogen or a hydroxyl group on that 2' position. And then the 3' position is where we're going to connect or link that nucleotide to the next one in sequence. And here's what that linkage looks like. Again, it's a phosphodiester linkage. So I'm going to draw that phosphodiester linkage, and then once I get to that oxygen of the phosphodiester linkage, I'm ready to essentially repeat that same pattern that I just had. So again, there's my nucleobase. We're going to have a hydrogen or an OH group, okay? And then we have an OH group that's there. So I'm going to highlight some things that could make this structure a little bit different, but again, the stuff that I'm drawing right now is always going to be the same. This is our 5' carbon. This is our 3' carbon. 5' carbon, 3' carbon. So when we refer to the directionality that we have for nucleic acids, we always say that they run 5' to 3'. And that's because we can sort of see here's the 5' carbon running to the 3', connected to the 5' to the next 3'. And they are connected. Again, this linkage that we have, I'm going to highlight it here in purple. This here is my phosphodiester bond. Okay, so this is my phosphodiester bond. So if you can draw this structure just like this, this represents a generic dinucleotide. Dinucleotide meaning that there's two nucleotides that are hooked together by a phosphodiester bond. But I'm going to give you a specific example here, and that's going to highlight how I can make different flavors of these for you to have to draw for me on an exam. So let's talk about an example. And you're going to see something that's going to be of this flavor. So you might see something that's this. P, D, G, P, C. I wanted to make sure I wrote exactly what I had. Now, what does this little code mean? Well, it's a dinucleotide, so you're always going to see letters here that refer to either A's, G's, C's, or T's, maybe a U if we're talking about an RNA, okay? But I see a G and a C there, okay? So that shows me that these are the bases. I've identified what the bases are going to be. And so on your generic structure, we're going to now map onto this specific example. You would just write the letter G there, and you'd write the letter C there. Now that seems pretty simple, but it is important that they go in the correct order, because the 5' most nucleotide is going to be that G. So that has to go here, and the C goes there. So I get that information from here. I'm going to highlight that this P, anytime you see a lowercase p, it refers to a phosphate. This again is referring to the phosphate that's between my G and C nucleotide. So that's the phosphate of the phosphodiester bond. Any other P's that you see are going to be either at the beginning or at the end. So any lowercase p's that you're going to see are going to represent P's that are between capital nucleotide letters, which represent phosphodiester bonds. So again, are only going to see one, because I'm going to ask you at most to draw a dinucleotide. But you could also see phosphates that are the first letter or the last letter, which are going to represent phosphates that are going to be either on the 5' or the 3' hydroxyl. You'll notice here that this represents what we call a 5' phosphate, which means at this position, we're going to have a phosphate. If you saw a P at the back end there, that would represent a phosphate at this position. So what does that really mean? I'm going to move this down just to give myself a little bit more room to draw this guy in. What this means is that at this 5' hydroxyl, it really is going to look like this. It's going to look like an oxygen that is now bound to a phosphate. Okay, so to highlight, this oxygen right here is that oxygen, and then it has a phosphate group. For right now, I want you to be able to draw this structure as is. I want you to show me what the structure of the phosphate is, and then be able to identify exactly where it's connected on this nucleotide. Okay, in future though, you'll be able to literally write this as an O and then a P. Okay, indicating we've got a phosphate group that's on there. Okay, or even you could just draw it as a P that we have there, with the understanding that we're going to have those 4 oxygens that's there. Okay, so for this time, I want you to be able to draw your nucleotide showing the full structure for the phosphate, but in future, we're always going to draw them as a P with a circle. Well, I know that was a lot on this slide, but this is getting us to sort of think about how do we draw the structure, both the monomer form, which is the nucleotides, and then the polymer form, which is a nucleic acid that's hooked together with a phosphodiester bond. So that's what we need to think about in terms of structure for nucleic acid. One last thing that I forgot to put on our example of our nucleotide here is I forgot to use some information about what this last letter was, right? I'm going to highlight it in green there, because that importantly is going to identify the nature of the sugar that we have. Whether it's a 2' hydrogen or a 2' hydroxyl group is going to indicate whether it's DNA or RNA. So if you see this D that we have there, that means that it's a deoxyribonucleic acid. If you don't see any letter there, the default is that it's a ribonucleic acid. It's RNA. Since we see the DNA there, I know that this group needs to be a hydrogen. So in my generic structure, I would replace that to say that that's a hydrogen that we have there. Okay, so that was one last piece that I forgot to put in here to make sure that we properly use all of the pieces of information from our specific structure to map on to our generic dinucleotide.